Hi, my name is Safura Reza and I'm in my 4th year of PhD under Professor Maciej Ugorski's supervision. So what you have to know about me is that I'm a total riddle nut. There's nothing I love more than figuring out something new and understanding something that wasn't clear before. And cancers are like the ultimate riddle, so of course I wanted to work with them. The big question for me is, why does a normal cell that's been living peacefully in the body suddenly turn into this monster that can spread all over the organism and survive crazy conditions like chemo or no oxygen? Because if we can figure that out, we'll know how to fight it. And these days scientists are really focused on how different proteins affect drug resistance in cancer. Proteins are pretty cool. You can isolate them and create them in the lab, so they're relatively easy to study. You can detect them with an antibody and so on. So naturally, I decided to not investigate them. In my doctorate, I focus on the role of lipids, specifically a tiny glycolipid called Galactosylceramide. Let me explain. If I had to compare a cancer cell to a Formula 1 car, then protein are the driver, super important. And glycolipids are concrete. In general, they seem to have a mainly structural role, but it turned out not to be the case. It has been shown that breast cancer cells that have a lot of this galactosylceramide or GalCer are definitively more resistant to chemotherapy and don't want to die. And you're going to tell me, yeah, great, but specifically, why do we need this information? In general, cancer science is not simple. There's always the same story. So we find this new protein that's involved in cancer, we're excited about it, but the bummer is we just can't block it to kill the cell, the cancer, because this protein is also in healthy cells and we don't want to harm those. But here's the thing, if cancer cells have too much of this GalCer and we block it, it won't kill the cancer cell or the healthy sister. It will just make them more sensitive to chemo, which could be a game changer in making treatment more effective. Like if you put the concrete out during a Formula 1 race, you don't stop the race, you just make the car go slower. And if you remember what I said before about understanding how it works lead to understanding how to fight it, well, in my thesis I showed that GalCer control a pathway of at least 5 proteins, and that's what caused this resistance to drugs. And get this, it turned out that galactosylceramide, which is on the cell membrane, activates the receptors for epithelial growth factor. This receptor is often found in large amount in different types of cancer like breast cancer. That's why different inhibitors are already used on it, which help in chemotherapy, but unfortunately they have a lot of side effects. We hope that now, knowing that GalCer can also increase the activation of this receptor, we'll be able to use a combination of inhibitor of GalCer together with inhibitor on epithelial growth factor receptor in the future. This could increase the effectiveness of the therapy while reducing side effects, helping hundreds of thousands of breast cancer patients every year. And to sum up, at the very end, I would like to quote J.R.R. Tolkien. Even the smallest person, in our case glycolipids, can change the course of history.